So, welcome to this class on Neuroscience of Human Movement. So, in this class we will continue our discussion on cerebellum. So, this is part 4 of our discussion on cerebellum. So, in this class we will be talking about recurrent loops that cerebellum has with cerebral cortex, inferior olive and Golgi cells. We briefly mentioned this in the previous class, we will discuss that with some details in this class. So, a major recurrent loop with the cerebral cortex. So, the cerebral cortex sends information to the cerebellum, not directly, it is not like that, but rather through pontine nucleus or through pons, right. So, through that and through that as shown here, right. So, pons serves as an important source of input to the lateral cerebellum. What do we mean by lateral cerebellum? Lateral cerebellum means cerebrocerebellum, is it not? That is the reason this region of the cerebellum is also called as cerebrocerebellum, right. So, the more medial regions are the spinocerebellum and the flocculonodular lobe is the vestibulocerebellum. We have seen this in the previous classes. So, and the output from the lateral cerebellum goes via the deep nuclei. So, actually not shown here is the deep cerebellar nuclei. In particular, what is the nucleus that is responsible for sending outputs to the cortex or processing information from the cerebellum, right. So, that is the dentate nucleus. So, from the lateral cerebellum projection is to the dentate nucleus, and from the dentate nucleus to the thalamus, more specifically to the ventrolateral nucleus of the thalamus. So, the thalamus then relays information back to the pre and primary motor areas. So, effectively completing a loop. So, the information starts from the cerebral cortex, from multiple regions of the cerebral cortex, and reaches the cerebellum via the pons, and that is processed in the cerebellar cortex, and via the dentate nucleus it reaches the ventrolateral thalamus, and from the thalamus back to the pre and primary motor cortex. So, this is one recurrent loop, and this is one of the prominent recurrent loops that the cerebellum has with the cerebral cortex. Then the other recurrent loop is with the inferior olive. We have said this inferior olive is one of the powerful or primary sources of input. So, there are two major sources of input to the cerebellum. These are massive fibers and climbing fibers. Massive fibers include all the inputs except inferior olive. This may be from pons, this may be from. So, inputs that originate from pons, input that originate from spinal cord and any other region are called massive fibers. Inputs that originate from inferior olive are called as climbing fibers. We saw what is the special feature of these climbing fibers. Climbing fibers climb through the granular layer. They do not make synapses with the granule cells, but rather they directly synapse with the Purkinje cells, make hundreds of synapses with with one Purkinje cell, for example, and in a powerful manner influence the output of the Purkinje cells. And we also saw that one climbing cell can innervate up to 10 Purkinje cells, but a given Purkinje cell receives input from only one climbing fiber, right. So, that information is a very powerful source of information is the climbing fiber, and it turns out that the deep cerebellar nuclei can also negatively or inhibit the inferior olive. So, in other words, the cerebellum can regulate the inputs that it receives, from where and what kind of information it is going to be receiving, right. And note also we discussed that inferior olive neurons are electrotonically coupled, or in other words there are gap junctions. Information communication within inferior olive between some groups of cells is electrotonic or electrical through gap junctions, right. This we discussed earlier. So, this involves another recurrent loop. Of course, then the output goes via the cerebellar nucleus to thalamus, etc. is the story that is not mentioned. Then the other recurrent loop is somewhat different from the previous loops. Note in the previous two cases, the recurrent loops were formed with structures outside the cerebellum. These were cerebral cortex and inferior olive. But this recurrent loop, the third one, is with Golgi cells, is situated entirely within the cerebellum. So, it turns out that the Golgi cells are inhibitory interneurons. So, these are inhibitory interneurons that are excited by the mossy parallel system, right. So, in the glomerulus they are excited by the mossy fiber, and parallel fibers also excite the Golgi cells, and in turn these Golgi cells inhibit the granule cells, right. What would be the purpose of such an arrangement is the question. Note that the number of let us remind ourselves that the number of granule cells is about 100 billion, we said that. So, if all of these cells are simultaneously active, it is not clear what is the meaning of that. So, because of this reason, it is required for us to have excitatory inputs from only specific granule cells that need to be active. So, that means there is a need to control which granule cells are firing when, etc. So, Golgi cells perform this crucial activity. What could be the effect of Golgi cell activity on granule cells? Well, there are two ways in which Golgi cells could affect the functioning of the granule cells. First is it could affect the magnitude of the total response. I will explain that in a minute, and it could affect the duration of activity of the response. So, when I say the response, obviously I am not talking about the response of individual granule cells, but rather when a bunch of granule cells are active simultaneously and some of them are inhibited, when say 10 granule cells are simultaneously active and say 4 of them are inhibited, then the sum total of their activity will reduce. So, when I say magnitude of the response will reduce, I am talking about the response or the total response of this system of the set of neurons under consideration. So, obviously individual action potentials will not reduce, their magnitude nor duration will not change. So, that is known. So, that is the stereotypical nature of action potentials that will continue for each individual cell. But when I turn off specific cells particularly by using Golgi cells, the total output from a set of neurons will reduce, or if I do not turn off, then it will increase. So, I could selectively modify the magnitude and duration of the response of multiple cells. One hypothesis about this function of Golgi cells is that in some sense the Golgi cells are encoding a form of sparse code, right. So, you want to keep a whole bunch of granule cells silent, and Golgi cells perform this crucial function. Only those granule cells that need to be active at a particular time are allowed to be active, keeping all the other cells relatively silent, because the number of granule cells is very large, you cannot keep them active simultaneously and respond to their activity. So, probably the Golgi cells encode some form of sparse code. This is an interesting hypothesis, continues to be discussed in the literature. So, in this class we have seen recurrent loops that the cerebellum has with cerebral cortex. This is via pons, the cerebellum as in the lateral cerebellum or the cerebrocerebellum, and VL thalamus via the deep nucleus or via the dentate nucleus. Okay. Then inferior olive cerebellum, it inhibits the inferior olive. So, cerebellum can control what kind of or from what kind of input it is going to receive and to what extent it can regulate its own inputs. Golgi cells regulate the function of granule cells. Okay. So, with this we come to the end of this class, and in future classes we will discuss vestibular cerebellum, spinocerebellum and cerebrocerebellum. So, thank you very much for your attention.